And without further ado, patient care, genetic testing in cancer patients, a case illustration with Cipriani, PharmD, BCOP, CPP, CGC, MS, and Jamie Richardson, BA, RN, BSN, OCN, and CCRP. So with biographical information, we don't have time to do all of this today, but I will say Amber Cipriani is a clinical pharmacist specialized in oncology care, pharmacogenomics. She currently serves as the Precision Medicine Pharmacy Coordinator at the University of North Carolina Medical Center, where she works to implement initiatives that improve medication and management through utilization of technology, genetics, and clinical decision support tools. Dr. Cipriani's position is a joint-funded position with the UNC Eshelman School of Pharmacy, where she serves as a clinical assistant professor delivering oncology pharmacotherapy content to professional PharmD students. Dr. Cipriani, well, what's one thing we should know that wasn't on that?
Yeah, hey Tim, I think your audio is breaking up, but I think what you're asking me is one thing everyone should know about me. I guess the recent thing in my life is my daughter just turned 18 months old, so besides coordinating things at the medical center, I'm trying to learn how to be a parent to a toddler. So that's the biggest thing going on for me right now.
Congratulations, that's outstanding. And I apologize, this is something odd with the internet today because it's saying it's great connectivity and clearly it's not. We also have Ashlyn Messmore, and Ms. Messmore earned her BA degree, highest departmental honors in Human Biology and Society Disability Studies minor at the University of California, Los Angeles in 2014. She then earned her MS degree in genetic counseling at the University of California, Irvine in 2016. And Ashlyn, what's something we should know about you that's not on the bio there?
Thanks, Tim. Yeah, something new in my life is that my 6-month-old started crawling yesterday. So I've heard that it's all downhill from here. But yes, that's something new in my life.
Wow. Well, that's exciting. Good deal. All right. And last, Jamie Richardson, BA, RN, BSN, OCN, CCRP, currently serves as nurse coordinator at the UNC Molecular Tumor Board, which assists clinicians in the interpretation of genetic variants to help guide patient care. Jamie, what about for you? Something we should know outside of your bio there?
Yeah, what most people don't know about me is that nursing is actually my second career. So my second time around. First was sports news. So I did a 180. Was what? News.
Okay. Wow. I did not know that. That's great. All right. I mentioned poll everywhere. Here's our first question. Genetic testing can be used to both guide cancer treatment and assess risk for cancer development. True or false? We try to make these a softball. I'll put that up in a moment. This activity has been planned and implemented under the sole supervision of the course directors in association with the UNC Office of Continuing Professional Development. Bill Wood, MD, MPH, and CPD staff have no relevant financial relationships with commercial interests as defined by the ACCME. Amber Cipriani, Ashlyn Messmore, and Jamie Richardson have no financial relationships with commercial interests as defined by the ACCME. And here's that poll. And we can see those that have answered it already seem to be trending on the true. And for our presenters today, how are they doing so far? Good. All right. Well, Jamie, I'll turn it over to you. Genetic testing in cancer patients, a case illustration.
All right. Thanks, Tim. You can go ahead to the next one. So I'll start by presenting the objectives that Amber, Ashlyn, and I hope to achieve by giving this talk. So the first is to define somatic and germline genetic testing and describe their role in cancer care. Second is to discuss the best ways to communicate genetic information to help bridge the language gap between providers and patients. And third is to explain the process of utilizing genetic testing to select optimal cancer treatment. You can go ahead. So we're going to start right off the bat with a case study. So we have a 46-year-old never smoking African-American man who presents to the ED with coughing up blood, shortness of breath. CT scan shows bilateral pulmonary nodules and what appear to be bone mets. He undergoes a same-day VATS, and pathology confirms lung adenocarcinoma with a PD-L1 of 5%, and a staging MRI unfortunately shows innumerable brain metastases. He starts carboplatin and pemetrexed inpatient, and when asked about his family cancer history, he states that his father was diagnosed with esophageal cancer at age 58, but he's not aware of other cancers in family members. Go ahead, Tim. And so the question that I hope to answer for you over the next several slides is what kind of genetic testing we would order for this patient. Go ahead, Tim. Anytime we talk about genetics, we should first touch on DNA. So this is a little bit of a refresher slide for those of you who maybe haven't thought about it in a while. So as a reminder, DNA is a molecule inside our cells that carries genetic info and instructions. It's packaged in chromosomes. Go ahead, Tim. It's made up of 4 building blocks called nucleotides, and those 4 are adenine, thymine, guanine, and cytosine. Go ahead, Tim. And if you're trying to explain this to patients, you can tell them that the sequence of these 4 letters, if you will, creates gene sentences and a story that instructs cells. So small changes in the orders of the letters can change the meaning of the sentences, and these changes could result in gene mutations. Tim, you can go ahead through the end. And it's also important to note that tumors have their own unique DNA profile, which leads us to the next slide and the concept of precision oncology. Okay. So the more we learn about cancer growth and behavior, the more it's become apparent that the disease can vary significantly from one patient to the next. So you could have 3 lung cancer patients, which all looks the same under the microscope. However, genetically, they're completely different. And that's where we come up with this notion that every patient's cancer is rare. Okay. Traditionally, treatment was based upon where in the body the cancer started. Go ahead, Tim. However, precision oncology enables us to detect unique genetic changes in the patient's cancer and then target them with treatments if we have them. So you could have one patient with breast cancer, another with prostate cancer, and a third with pancreatic cancer, who are all taking the same medication because all of their tumors harbor the same genetic mutation. Okay. Over the past 2 decades, the use of targeted therapies has increased exponentially. And this kind of therapy is the foundation of precision oncology. Next, Tim. So Amber's going to go over targeted therapy in much more detail, but just to give you kind of a high-level view, these are treatments which interfere with specific cellular processes used by cancer to grow and spread, and this is different than chemotherapy, which targets all rapidly dividing cells. Go ahead, Tim. Some examples you might be familiar with are monoclonal antibodies, small molecule inhibitors, and antibody-drug conjugates. Breast cancer and chronic myelogenous leukemia actually had some of the first targeted therapies. So we have trastuzumab or Herceptin, which was FDA approved in 1998 to target HER2 gene overexpression in breast cancer, and then imatinib or Gleevec, which was approved in 2001 to target the BCR-ABL fusion gene in CML. And what you see on the right is the cover of Time magazine from May 2001. And this is one of the earliest times that the mainstream media really embraced this concept of targeted therapy. Okay. Genetic testing is a pretty broad term, but at the heart of it, what we're trying to figure out is if a patient either has a somatic or germline mutation. So somatic mutations are those that occur in the tumor only. This is the opposite of germline mutations, which are present in egg or sperm and therefore in all cells of the body. They can be inherited, and they can even cause family cancer syndromes. And Ashlyn's going to talk more about germline mutations. Go ahead, Tim. As far as somatic testing, you may have heard the terms molecular profiling, tumor sequencing, but typically what folks are referring to these days is something called next generation sequencing or NGS. And this is a technology that can process actually millions of DNA sequences in parallel to look for gene variants in cancer. And this is in contrast to older generation sequencing, such as Sanger sequencing, which was only able to look at smaller DNA fragments one at a time. So this high throughput method gives us a lot more information a lot quicker. Go ahead, Tim. If you're trying to explain this to your patients, you can just tell them to think of it as a very fancy spell checker for tumor DNA. And some of the NGS labs that we use here at UNC, Foundation Medicine, Guardant, NeoGenomics, and more recently Tempus. If you are interested in learning more about each of these tests, you can Google the NIH GTR or Genetic Testing Registry, and they go into each one of these in detail. And then that picture at the bottom, that's just that's a picture of what one of these sequencers looks like. When is genetic testing appropriate? So this could actually be a lecture all on its own, but just to highlight a few key points. So somatic testing is most often used to guide treatment in cancers with common genetic variants. For example, in about 15 to 30% of non-small cell lung cancer patients is something called an EGFR mutation. So if patients have specific EGFR mutations, there are FDA-approved targeted therapies for them to take, which were shown to be more efficacious in this population up front compared to standard chemo. We also might order it when a patient is out of standard treatment options or they cannot tolerate those options. It could be required as part of eligibility for clinical trial participation. And we sometimes use it to confirm a cancer diagnosis. And a good example of this is in sarcoma. Not only are these cancers rare, but their subtypes tend to overlap histologically. So this can help us nail down a diagnosis. A few key scenarios for germline. So a young age at diagnosis, such as a 30-year-old breast cancer patient, or a strong family history of certain cancers. Nowadays, it's done up front for certain cancer types, such as epithelial ovarian cancer or newly diagnosed stage 4 prostate cancer. Sometimes ancestry is associated with certain genetic mutations in cancers, and one of the most well-known is Ashkenazi Jewish heritage and BRCA mutations. And then it's possible that we found a pathogenic variant by somatic testing that looks suspicious for germline testing, and then we can proceed with that and see how it turns out. Go ahead, Tim. A few additional things to keep in mind. Both somatic and germline testing are referenced in NCCN Guidelines. For those of you not familiar, the National Comprehensive Cancer Network is a professional alliance made up of about 30 cancer centers across the country, and these guidelines are used for clinical policy, clinical practice, payer reimbursement, those sorts of things. Also good to know that the treating medical oncologist, can we go back, thanks. Also good to know that the treating medical oncologist will most often be the one to order somatic testing because it's their treatment plan that will be affected. However, for some patients who need this testing done right away to even start treatment, you might see a surgical oncologist, for example, order it. And then last but not least, if you suspect an inherited predisposition to cancer based on age, ancestry, or something else, the patient should really be referred to genetic counseling first, not only to understand the purpose of germline testing, but the potential impact on them and their family members. All right, so back to our case study. If you remember, a 46-year-old gentleman with stage 4 non-small cell lung cancer and a limited family cancer history. So the NCCN guidelines for non-small cell lung cancer are about 200 pages long. So this is just a snapshot for our purposes. Go ahead, Tim. So if you can see on the left-hand side under clinical presentation, so we said in the beginning he had metastatic disease. And then in the middle, under histologic subtype, pathology confirmed adenocarcinoma. And on the right, you see the recommendations for biomarker testing. So they say molecular testing for that EGFR mutation we talked about, as well as ALK, ROS1, BRAF, NTRK, MET, RET, PD-L1, etc. But if you look at the orange arrow, it says that testing should be conducted as part of broad molecular profiling, or NGS, if you will. So that leads us to our first poll question.
All right, and we're having a little trouble activating that poll. Bear with us and we'll see if we can get that to, there we go. So what kind of genetic testing would be ordered for this patient? None, based on the patient's age, NSCLC diagnosis, stage, and family cancer history. And Jamie, are you still getting the audio breakup there?
You sound better.
I sound better. Okay,
Yeah.
I'll keep reading. B would be somatic testing based on NSCLC diagnosis and stage. C is germline testing based on patient's age, NSCLC diagnosis, stage, and family cancer history. Or D, germline testing based on patient's age, NSCLC diagnosis, stage, and family cancer history. Jamie, how are they doing?
So they're doing pretty awesome.
Good.
So the answer is B, somatic testing. I see a few people chose D in the beginning, and I don't know the exact reason, but I mean, being 46, he is about 20 years younger than the average age for a cancer diagnosis in the U.S. So if that was the thinking, he's not quite young enough for us to think germline right off the bat. And then if you're thinking about his family cancer history, he doesn't seem to have an extensive family cancer history. He was only aware of one family member, his dad. So the correct answer at the end of the day would be B, somatic testing. All right, so we know that our patient is going to have to undergo somatic testing. So how might we talk to him about this, given that he already has a ton on his plate with a new cancer diagnosis, and this stuff can be pretty complicated. So these are some of the things we can tell him. We can say that his doctor can order tests on his tumor tissue or blood to look for targets or traits that could be causing his cancer to grow. Go ahead, Tim. You can put all of them, Tim, if you want. And that testing may find targets that his doctor can treat with a drug. However, that not all of those options will be FDA-approved drugs, and they could only include those available on research studies, which would be important to mention, especially if you're seeing patients in an academic center. We would also want him to know that testing could find targets that don't yet have a drug to treat them because we're still learning about them. And then in some cases, testing isn't able to find any targets to treat with a drug, unfortunately. But we can reassure him that once his doctor has his test results, that they'll work together to find a treatment that works best for him. All right, so he got somatic testing through Foundation Medicine, and these are the results. So if you see the gray box at the top, it says no reportable alterations with companion diagnostic claims. And what this essentially means is that in his specific tumor, there were no genetic variants, mutations, etc., that currently have a corresponding FDA-approved therapy. However, further down the page, there's a section called Other Alterations. And there's also a little disclaimer saying that these results are not prescriptive or conclusive for any specific therapies. However, they are present in the tumor and they could become important down the road. And those are all listed in the blue box. And you can imagine if you're a clinician who is ordering this kind of testing on a lot of patients, this is a lot of data to be getting back for each patient and a lot of information you have to sift through. Luckily at UNC, we have something called a molecular tumor board, which Amber will talk to you a little bit more about. And we can kind of take some of the onus off the providers and do their homework for them and look into these alterations. So that's the case for this gentleman. He was referred to us at Tumor Board. Tim, you can go to the next one. And after taking a preliminary look at his testing results, we recognize that that BRCA splice site mutation there that has the arrow next to it is actually a known pathogenic germline mutation. All right, so we reached out to Foundation Medicine to ask a few more questions about the test. And one thing we specifically asked them for was the variant allele frequency of this mutation. And what that is the percent of tumor cells that they looked at that carry the mutation. So they came back to us with 77%, and a variant is potentially a germline mutation if this VAF is somewhere between 50 and 100%. So because his was right smack in the middle, we made the recommendation to refer him for genetic counseling and testing while we looked into his other variants. And this is where I hand it over to Ashlyn.
Thank you. So as a genetic counselor, when we receive information on patients, we look it over and then we're able to actually spend an hour with each of our patients going through their family history, their personal history, and some other risk factors. All of that combined helps us figure out where is our suspicion level that this could be actually an inherited cancer predisposition syndrome. If it's very high or if it's very low, we're able to talk with our patient about that. And in our discussion, we talk about the testing options. We actually tailor our testing to our patient. And we also get to discuss types of results, the risks that could come along, as well as the surveillance that we can offer, insurance implications, which we get a lot of questions about that, as well as privacy. And we talk about some laws that have been passed to protect us, and reproductive implications. All that together lets our patients make an informed decision about proceeding with testing. Next. This is actually an example of a family history. It's not one that I've drawn, but it is helpful just to see. If you click the next slide really quick, the generation lines show us sort of who is in the family and how is everybody related. Each of these lines tells us that, for example, Jeff on the all the way right and Mary Ann, they're siblings. They share the same parents. Marianne and Michael, who's right next door to her, they're connected through a horizontal line, and then below them are their progeny. So those are their kids, and that's how those generation lines work in a family history. Next slide. To orient you a little bit, these circles are female, and then the squares we draw are for men. And everyone that genetics has seen at UNC has a pedigree that's been drawn for them. More recently, we're actually uploading them in Epic under media. So if you ever need to see a pedigree for someone who's seen genetics, I would check there. Next slide. This is an arrow that helps us tell who the proband is. The proband is sometimes our patient, but the proband is actually sometimes not our patient. The proband is whoever initially led the family or the person to medical attention. So if I have a patient who's a daughter of someone who carries a BRCA2 gene, the daughter is my patient, but the proband is actually her mom. Next slide. Okay, so how do I explain the difference between kind of somatic and germline testing? I basically start out by saying that we all want to be Oprah, obviously, but Oprah in this photo here, she has her two hands up in the air. And what that tells me and what I explain to patients is that someone with these open hands, I say that an open hand means that their particular gene, like Jamie mentioned before, that's designed to help protect them from cancer is working great. You get one from mom and one from dad. And I actually do this hand thing because it really helps. And so when I talk with patients, I say, okay, most folks are born with a working copy from mom and a working copy from dad. And then I get a lot of pushback like, well, if my genes are working, why do I have cancer? And I say, that's a great question. Honestly, because during our lifetime, there's enough somatic changes. I call it wonkiness. So there's enough wonky changes in our genes for them to actually stop working. And then cancer is sort of allowed to grow and develop. And it's through nobody's fault. Honestly, our genes are only designed to help, our body for as long as they can, but we're exposed to all sorts of stuff in our environment, like the sun or smoking, or just the natural aging process kind of knocks out their function. So midway through our life, one copy usually stops working, but this backup one kind of helps out and can get you through. Or it could also stop working. And when both are kind of not working as well as they should, then we can see cancer grow and develop in a person eventually. That is someone who's born with two perfectly working copies working great, and eventually they can develop cancer later on. That's nothing that they inherited. Someone who inherited a non-working gene is like the picture on the right. They don't look like Oprah. They look like this. And that person could then pass down that non-working gene to their children. And that person only has sort of one copy designed to protect them. And that is what that increased risk for cancer comes from, because eventually, potentially, this copy could also stop working. And then they're left with two non-working copies really early on in life. And that's where that increased risk for cancer can come in. So that's how I explain the difference between somatic, you develop it over time, and germline. You're actually born with one of these genes already not working in every cell. And that's what you can pass down to the next generation. Okay, next slide. So what happens when we get the results? We have three different options. We have pathogenic, nothing, or a variant of uncertain significance. I think of these different results like differences on our recipe. So a pathogenic variant, I always talk about it as if we were baking a cake and the cake recipe called for 2 cups of sugar. You would probably put in about 2 cups of sugar. That would be our normal result. If instead that recipe that we were reading through said 20 cups of sugar, that's very different. And that's probably not what you intended to make. That would be a pathogenic variant, something that's very obviously different about that recipe or that gene. A variant of uncertain significance is a little bit different. It's more like substituting sugar for Splenda. Could I tell the difference? No, I'd probably eat the cake. But could someone with a far more refined palate tell the difference? Maybe. So this is where we get sometimes this is important, but most of the time they're not important. So if we find a VUS, we do not alter medical management at all. That's hugely important. Next slide. So I also want to go through examples of results. Next slide. These 2 examples are ones that I'll quickly go through. All the way on the left, as you can tell, the gene name is written in italics, so ATM and BRCA2. Next. What's helpful to know is this C dot. A lot of these results seem like gibberish at the beginning, but they're not. They're easy as long as you can kind of explain it in a way that makes sense to a patient. So the C dot tells me that we're going to chat about the nucleotides, or like Jamie mentioned, the sequence of letters that makes the gene make sense. This particular one, this first one in ATM, it says C.486 del A. What I know is that C dot means we're talking about the letters that make up the gene. The 486th letter was supposed to be an A, but the fact that it says del means deletion. So I know that that one A is missing. A lot of patients will say, okay, it's just one letter. How could that possibly change my body? Well, the problem is we read these codes 3 letters at a time to make an amino acid and make sense of it. When you delete one of those letters, our body keeps reading, everything just gets shoved over. So examples, the red dog ran is a bunch of 3-letter words, and that makes sense. But when we delete the E, for example, in red, it becomes the redida ogren, which is great, but it doesn't make any sense. And that's the same thing that happens in our body. Next, please. This changes the amino acid. Next, please. And this is like a chart that will help, because sometimes labs do a 3-letter code, sometimes they do a 1-letter code for an amino acid, sometimes they write out the whole thing. But this is how we can tell that an amino acid looks different. Next slide, please. So then what happens? So we changed an amino acid. It should have been glycine, now it's histidine. Well, that actually matters because this frame shift, so moving everything over, resulted in a stop, meaning our body stopped reading the recipe. And that's what led to the change in the gene. Next slide. The BRCA2 example is a little bit different. There's a substitution instead. So C.120C to T is what that means. Usually there's a C in that spot, but we see a T instead. There's also deletions, insertions. Next slide. But why this is helpful is because it helps us understand the result a little bit better. So this is the poll question. Which statement accurately describes this report? This is just a sample report. It's not our patient. It's just one that tells us about this particular gene. So what do we see? A VUS in the CHEK2 gene showing a protein change, a pathogenic mutation in the CHEK2 gene, a VUS in the BRCA1 gene, or a VUS found in CHEK2 showing a protein change, and this patient now qualifies for increased cancer screening.
All right, and as always, we want to say thank you to everyone for jumping in and responding. Take about 5 more seconds, if you would. Ashlyn, how are they doing?
They're doing really well. I'm glad. Really glad. So based on this, we know that it is a variant of unknown significance. That's true in the CHEK2 gene. Some folks said the BRCA1, though BRCA1 is listed in the title of the report, the results only talk about CHEK2. And since it's just a variant of unknown significance, we're not going to alter any medical management because the vast majority of these, again, are nothing. So A is the correct answer. Next slide. How do I explain this germline result to a patient? So I just told you the C dot and all of this good stuff. So our particular patient had a germline BRCA1 mutation identified when we got to see them. This C.213 minus 11 T to G is a little bit more complicated, but not terribly so. The minus 11 lets me know that this is in an intron, and that tells me that it's something where it's before the exon, and the exon is usually what we read when we think about reading through our genes. I think of it like a magazine. So an intron is like the advertisement beforehand. Usually you can rip those out and nobody cares because you want the articles in the magazine and you don't care about the advertisement. This particular change in the intron is like it's found in one of the advertisement pages. So it's a little bit different, but if you ever had to explain it, just use a magazine example. It's found in one of the advertisement pages, but it really matters. Next slide. Because now we know that there's a germline variant, we would potentially need to test all living first-degree relatives and then more depending on which side of the family this comes from. If this is ever the case and you need to locate a genetic counselor near a patient, this website is wonderful. It's nsgc.org. You can just type in your zip code and find the nearest genetics group or center to your patient. And it's really user-friendly, and we recommend it highly to find a qualified genetics person near them. Next slide. I get a lot of questions about the difference between somatic and germline, and do I really need to do both? I think this is a great article if you have some time to read. It talks about what somatic testing can miss. And believe it or not, it can miss about 8% of germline variants simply because of the way the technology works and because we're testing tumor cells that can delete genes or have wonky changes that get missed. And it also talks about the repercussions of missing these germline variants, which can lead to inadequate patient care and new primaries that could have potentially been prevented or avoided or at least been better surveilled. I appreciate it. I'm going to now pass it to Dr. Ambry Cipriani to continue.
Thanks, Ashlyn. Thanks, Tim, for advancing the slides. So just kind of a recap. So Jamie kind of gave you an introduction to genetic testing and talked about sort of the difference in somatic and germline testing and when we might order somatic testing in a cancer patient. And then Ashlyn just talked about germline testing specifically, which I think most people would sort of summarize this as, you know, we do germline testing. We're really trying to identify patients who have an inherited risk of cancer so that we can sort of implement early screening or early diagnoses techniques in those patients. In the third part, I want to give you a little bit of a flavor and keeping with our case of how we might use genetic testing to help guide our treatment approach to patients. And a lot of people sort of think of this as precision oncology. Jamie used that term, or precision medicine, which that term is in my job title. And we're focusing on genetics, but I also just didn't want to lose sight of precision medicine is kind of a very broad practice. And so this definition of precision medicine I lifted from when in 2016 President Obama introduced the Precision Medicine Initiative. And so using that definition, it's an emerging approach for disease treatment and prevention. So when we talk about cancer, we think of both. That takes into account individual variability in genetics, but also in microbiomes, environment, and lifestyle. So I think we're trying to give you tools in this presentation to interpret the genetics, but I also just want to emphasize that genetics is just a piece of the puzzle that we're trying to solve, which is how to best care for that individual. So Tim, thank you. So how do we use genetic testing to guide treatment decisions, specifically in cancer? And so I put it sort of in 4 buckets. And when I say genetic testing, I'm sort of referring to both germline and somatic, although I'll say right now in oncology, we're relying a little more heavily on the somatic testing because that's the tumor genes that we're worried about. So we can use genetic testing, and Jamie mentioned this, to be diagnostic. So if we are having trouble coming up with a histologic diagnosis based on what the tissue looks like under a microscope, sometimes genetic testing can help us figure out what actual cancer type this is. We can also use genetic testing to help us determine prognosis. And so genetic tests have helped clinicians determine the aggressiveness of a person's cancer, and that can help us better enroll patients in clinical trials and sort of stratify who just at baseline, regardless of treatment, is going to have a different path in their cancer journey. And then genetic testing, I think the one I think about most being a pharmacist can be used as a therapeutic biomarker. So specific genetic variants can be used to either predict efficacy from a given drug or treatment, and they can also be used to predict resistance from a given drug or treatment. And then the final one that we won't go into today, but I just want to emphasize, is that genetic tests can also guide drug dosing. And so there are many examples of pharmacogenomics, meaning testing germline inherited mutations in different drug metabolizing enzymes, and understanding how those drug metabolizing enzymes might work better or worse than the population in general, and using that information to determine a better dose for patients. And so the one example I have here is genetic screening of this gene called UGT1A1. Patients with a particular inherited genotype in this gene might require a dose reduction of irinotecan to minimize their risk of severe neutropenia. So Tim, if you hit the button again, there's a big, beautiful red circle. So I'm going to focus on genetic testing and how we use it in guiding therapy. But I just wanted to make sure you understood it's used for a number of things. So next slide, if you will. So when I think of genetic testing for therapeutics, I think of tests that might predict efficacy and resistance. And so why do we really care about this? Why do we need to do genetic testing to predict efficacy? And so I think some of my my studies on here are landmark studies because they're old now, which ages me, but these are landmark studies because what they're looking at is what's the difference between targeted therapy, or these therapies that maybe are specific to patients who have certain genetic variants, versus cytotoxic chemotherapy, which, like Jamie mentioned, worked for certain cancers, but it really was pretty non-specific. It targeted cells that were dividing. The tumor cells hopefully are dividing quickly, the quickest, but they had significant toxicity on normal cells. And so this is a study looking at patients who had EGFR mutations and lung cancer, and comparing their treatment response when we give them cytotoxic chemotherapy, which is carboplatin and gemcitabine, compared to if we give them a targeted drug, or erlotinib. The chemotherapy, carboplatin and gemcitabine, is IV, or erlotinib is oral, so there's a convenience factor, and the toxicity profiles are different. And what you can see here is that with old cytotoxic chemotherapy, our progression-free survival was 4.6 months, And when we gave patients the targeted therapy, we almost tripled our progression-free survival in this patient population. Not to mention they could take the drug orally and have different, some might argue, easier to tolerate toxicities with erlotinib. And so next slide, Tim. And then when we think about therapeutic markers of resistance, I think this is equally important. There are fewer examples of this, but I think it's something that we're thinking about more and more. So this is just an example. So treatment with this drug cetuximab, which is an EGFR targeted monoclonal antibody. If you just looked at sort of this group of patients included in the study with colorectal cancer, it did demonstrate efficacy in patients who were refractory to chemotherapy. So we thought, okay, everyone should get cetuximab. And then when we looked closer at these colorectal cancer patients, we realized that some patients responded really well, and then there was this subset of patients who didn't respond at all. And there was some efforts to try and figure out why that was. One of those efforts included genetic screening of those tumor samples. And what they found is if patients have these mutations in a protein called KRAS, none of them got any benefit from the cetuximab. And so what I always tell patients is that, you know, you're not getting any benefit. I'm just giving you a big bill and a lot of side effects. And I don't want to do that as a pharmacist or as a clinician. And so appreciating these markers of resistance are equally as important as we learn more about genetics and cancer. So next slide, Tim. So how do we sort of identify these therapeutic biomarkers? Well, it is with sort of wide-scale screening of tumors. And so this graph I have on here is an example. It's data from a cohort of patients included in this MSK impact trial. And so this particular pie chart is looking at over 5,000 patients who had non-small cell lung cancer and their tumors were genotyped. And what we found is that there were patterns as far as what genes were mutated in lung cancer. And so we're using these patterns to identify therapeutic biomarkers and develop drugs that we think might have efficacy in these different cancer types. And this pie chart is going to look different depending on the tumor type. But for example, in non-small cell lung cancer, in this cohort, about 30% had mutations in KRAS, about 30% in EGFR. And we've talked about EGFR inhibitors, but I also want to just bring your attention to the other genes listed on here. So for example, BRAF, in about 5%, I actually have drugs that can target specific BRAF mutations. MET is another gene we see mutated in lung cancer. I have drugs to target that. ALK, guess what? I have a drug to target. ROS1, guess what? I have a drug to target. I couldn't say this 10 years ago, but as we get more and more drugs in the pipeline, we're able to target these biomarkers that we've seen mutated in cancers, and our armory is getting bigger. All right, next slide. So this is just to give you an idea. Now there are actually sometimes multiple options to treat a given gene. So this is looking at first-line options for mutated EGFR. And what I want to draw your attention to or just emphasize is that just because someone has a mutation in a gene does not mean that a given drug is going to work for that mutation. And so that's sort of the first step in the process when we're evaluating these drugs and choosing therapy. Number one is figuring out, is it actually what we call pathogenic or oncogenic? So do we think it's driving or contributing to the tumor cell dividing and being a bad, nasty cancer? And then the second question we ask ourselves is whatever drug we're considering actually active, or does it have clinical efficacy against that particular mutation? And so you can see I have this list of EGFR inhibitors on this slide, but the FDA approvals for them are slightly different. So all of the, or most of them have this exon 19 deletion and L858R mutation. But then for afatinib, you see the approval just says non-resistant EGFR mutations. And it's kind of left up to you to figure out what that means. And I'm going to show you how to do that in a second. The other thing I want to mention for this slide, and that's okay that you've advanced it, Tim, is just that these drugs can have slightly different toxicities. So remember, we talked about precision medicine. Don't forget about your actual patient. If you have the good fortune to be choosing between drugs, we still do things like consider organ function, adherence, cost, and toxicity profile when choosing. Next slide. So this is just to emphasize, this is data from June because Jamie and I were commiserating. It's actually not collated anywhere how many drugs have genetic information in their labels. We had to actually count this, and I didn't want to count again. But as of June, when I checked and actually counted, there were 164 oncology-related genetic biomarkers included in FDA-approved product labeling. And then if you look at the 37 tests that the FDA has approved as companion diagnostics, 35 of those are oncology related. And so this is just a huge booming area in oncology that's going to continue to grow. Next slide. So getting back to our patient, and I want to leave some time for our audience response question and questions, but you're probably looking again at this report, and it probably still looks like alphabet soup to you, and I just want to reassure you that most days it still looks like alphabet soup to me. It takes a lot of practice to figure out how to interpret this report, but the approach that we generally take is, as Jamie mentioned, we're first going to look to see if there's anything reported in this FDA-approved or companion diagnostic claims section. And then when you're looking at a long list of variants, it generally starts to stand out to you which targets have drugs approved for them. The other thing I look for are oncogenes versus tumor suppressor genes. We tend to have more targeted therapy for oncogenes as compared to tumor suppressor genes, although that's changing. And so, you know, the approach with this patient when I look at this report is I'm drawn to the BRCA1 for reasons of that could be germline. There's also some data in other cancer types to support use of targeted agents if they have this mutation. And then obviously this is a lung cancer patient, and so I sort of railed on you the prevalence of EGFR mutations. This particular mutation was not on that list of FDA approvals, and so our goal here would be to figure out if this mutation is associated with response from the EGFR inhibitors. I will tell you the other mutations on here, obviously we reviewed them. They are in tumor suppressors, and there's no targeted therapies associated with them, and I'll show you how to look that up for yourself in a second. Next slide. So you're probably thinking this all sounds great, but how the heck am I supposed to figure this out on my own? And so some helpful things as far as process and places to look. The first thing is we're showing you a snapshot of the laboratory reports from the next-gen sequencing for this patient, but the actual reports tend to be a lot longer, and they do include information to help you interpret the variants, meaning is it pathogenic or oncogenic like I mentioned, or are they not sure? And it also can have information about clinical trials or targeted drugs that may be efficacious for that given variant. So that's a good place to start. Jamie brought up the Nccn Guidelines, and I didn't mention those will help you figure out if you should do genetic testing. They will also help you figure out if there's evolving data to support targeted therapies for a given genetic variant. The drug package inserts can also be helpful. So if you have an EGFR mutation and you don't know it's targetable, one way to approach it would be to look at the approved EGFR inhibitors and see if you can find that particular mutation in the package inserts. And then the 3 websites I have listed here are sort of knowledge bases for precision oncology. And so in developing this presentation, we actually had some discussion about which one we thought was best to use. It's just like everything, how everyone has their favorite. My favorite is this OncoKB website, but the other 2 are useful and just formatted in a slightly different way. So I wanted us to sort of practice with this case using OncoKB. So if you're online shopping or doing something else on your phone, take a break and pull up this OncoKB website for me. It's easy if you just Google it, it should pull up by itself. And what I want to do, Tim, I think if you advance the slide, is using that OncoKB website, I want you to see if you can just figure it out. I'm throwing you to the wolves. Which EGFR inhibitor you might recommend to treat this patient. can you let us know? Oh, sorry, I was reading the chat. Let's see. And I know this might take you a minute, so Tim, I know we should still be able to finish with a few minutes for questions. And as you're doing this, I'll just talk you through, you know, when you pull up the website, first it's going to ask you to search by gene. And so in this case, you should search EGFR, which is the gene. And then you should be able to pull up a page that gives you quite a bit of information. And there's actually a search box to your right where you should be able to maybe type in this particular variant. So I don't know how many more responses you think we're going to get, Tim.
Yeah, we'll wait another 15 seconds or so. And Amber, I want to applaud you. You've added a new level of interactivity to our interactive questions. I love this. This is really great to have people interacting with a site in order to be able to provide the answers. That is great. All right, in the interest of time, you want to go ahead and let them know how they're doing?
Yes, and I love the change in poll everywhere. So at the beginning, osimertinib was winning, which if I had my choice without knowing any of the data, I'd rather pick osimertinib too because it has less side effects. But if you actually pull up this variant on OncoKB, you'll see there's actually more evidence reported in the literature for the drug erlotinib. And so I think that's why we're seeing the change, because people are actually looking in the literature and figuring out based on this variant, which is awesome. So Tim, if you go to the next slide, I just want to draw your attention to how you can incorporate this step in your process to help get you started if you have questions about you know, why you would choose one drug over the other. And so if you look on this OncoKB website, I encourage you to spend more time with it. But it does give you the level of evidence. So this is giving you 2 EGFR inhibitors, erlotinib and afatinib. The lower the number, the higher the level of evidence. So in this case, there's more evidence to use erlotinib, but there is some evidence to use afatinib for this particular variant. And then I couldn't show you in real time, but if you hover over that citations number, it will actually pull up the specific, whether they be case series, case reports, or actual clinical trials that support this claim in OncoKB. And so that can be a really good place to start your reading if you want to learn more about it. So again, this is not the only resource, but if you're in a bind and you want to learn more, I feel like this is one resource that you can kind of hopefully come away from this presentation knowing how to use. Next slide. And then I'm running short on time, so I'm going to just let you look at this in the handout, but I wanted to emphasize at UNC we have a great molecular tumor board that takes referrals within the UNC system to help providers sort of navigate some of these more challenging cases. Next slide. And then this lecture, we wanted to focus too on how to discuss therapy with patients. My biggest takeaway when I'm discussing targeted therapy with patients is tailor the discussion to the individual. So think about their health literacy, baseline knowledge, and their experience with cancer. The discussion's probably going to occur over multiple visits. So I like to focus on the need to know if I'm talking about a drug, focusing on how to take it and side effects, and then trying to build that understanding of why we're using that drug for a given genetic variant over multiple visits. And then when you're talking about targeted therapy, it's important to compare and contrast treatments and consider patients' personal experience. So I have this picture of this young girl with I guess, cancer on this slide because this is the patient that most people picture when they think about cancer treatment. They think about losing their hair and throwing up. And as we talk about these targeted therapies, that tends to not be the patient experience. And so I want to make sure I'm addressing any anxieties or pre-existing expectations to treatment for patients. Next slide. So I'm going to conclude sort of for all of us. So I want you to know recommendations for genetic testing, they're going to vary among the different cancer types and with different clinical scenarios. Although the role of both somatic and germline testing is continuing to expand, in both solid and malignant hematologic cancers. The other takeaway is that genetic testing doesn't always hold all the answers. And so I think with this presentation, we gave you a case where we had a targeted therapy that we could treat the patient with, but that's not always the case. And so helping patients understand that up front is really important. And then all members of the healthcare team are going to play a really important role in ensuring patients understand their treatment. And so hopefully using some of the verbiage that Ashlyn uses can help you better explain genetic testing to patients. With that, we'll take any questions. I know we are right at the end, so I apologize for that.
All right. Thank you all so much. I'm going to keep my video muted just to try and preserve the integrity of my audio with the limited bandwidth I've got here at my site today. So this is your opportunity to ask questions, so please go ahead and submit those, and we'll take those and promote those as soon as we get those. And what I'm going to do while we're waiting, oh, we've already got one that arrived. Good. Well, it says good presentation, wonderful information. Thanks so much. So we've got a few that are showing up here. While we're waiting for questions to come in, I am going to go ahead and go over a few things I need to wrap up, and then we'll come back to the question page. As always, we want to thank the citizens of North Carolina for making this possible through the General Assembly and their generous support of the University Cancer Research Fund. We want to thank the UNC Lineberger Comprehensive Cancer Center and the team here at the UNC Lineberger Cancer Network, Mary King, Veneranda Obore, and John Powell, for all the hard work they do for each and every one of these presentations.